Here are few tools which are available for the pharmacophore modeling and the QSAR thing, the PharmaGist, Zinc Pharma. These are the freely available. This build QSAR is not freely available basically. This PharmaGist, Zinc Pharma, PharmMapper, these are the freely available tools. Okay. QSAR toolbox is something that will do everything for you. You just have to, it need to be buy, but it will do everything for you. It will give the from the pharmacophore to the final QSR model, it will give to you. Okay. So till here the LBDD was done. I hope LBDD is clear. Okay, how can we use machine learning approach to build a QSR? See, whatever we have discussed, that is the machine learning only. The model we are creating, okay, the selection of the data, that all the part of machine learning only. Machine learning is what? In machine learning also we use the regression, the relationship basically. Whatever the things you are collecting, whatever the things you are considering, that should be related to each other. That is what the machine learning is. What is machine learning? You are preparing your machine, right? Here also we are preparing the model, the QSR model, which we are saying. Further, the new compounds will be checked with this QSR. That is the machine learning itself. So there is okay. Then comes the structure-based drug designing. Okay. In that case, in the previous thing we have done, like the structure is not there. Okay, so we have started working on the ligands. That why it was the ligand based drug designing. Now the structure based drug designing, that is the widely used one. Okay, the structure is available. Okay, as I told in the beginning, if the structure is not available, we can go for the modeling also. We can model the structure. For that also the tools are available. Okay. But if the crystal structure is available, that is very good thing, because that those are highly accurate than the modeled one. Okay. And when we talk about the 3D structure, the 3D structure can be generated using the X-ray crystallography, NMR spectroscopy, and homology modeling. Basically, X-ray and NMR, these are the experimental method. Homology modeling, that we bioinformatician, we computation biologists do. Okay. While understanding the cycle of the drug designing, what we have to look for, we have to inhibit the target, we have to enhance the activity, okay, and we have to go for the compounds which are complementary to the active site. These are the compounds we have to look for, which inhibit the target, which either enhance the activity of the target, or which is complementary to the active site. These are the things we consider. And what is the biological target? When we talk about the biological target, it could be any protein, nucleic acid, anything. Okay, it should be involved in the pathway, in the metabolic or the signaling pathway. It should cause the disease. It should be responsible for the pathogenicity, either survival of the pathogen or whatever. Or it should be infectious to the disease. Okay, it should be infective. Okay. While combining these two, the compound and the biological target, okay, that is the part of your SPDD. When we get these two and we identify some compound, this compound, okay, this compound will go to the preclinical trials, then the clinical trials. This is not in our hand, okay, that is completely different procedure. And then we go for the drugs. Now, whatever the drug we are going for, whatever the compound that will become the drug, that should be safe, that should be efficient, that should be stable, soluble in your body, and that should be novel. While launching the drug in the market, these are the things that should be there. Okay. Here we will discuss brief basic terminology. Okay. We have started with the target and the ligands. Okay. So target is what? The protein structure which we have to target. Ligands are what? The chemical compound which we will be screening. Okay. So while screening the compounds, we start with the 1,000 compounds. Out of those 1,000 compounds, while screening, we identify only 100 compounds are those which are binding to the protein. So now these 100 compounds will be the hits. Okay, they are binding to the protein. 900 compounds will be filtered out. Next comes, out of these 100 compounds, next we have to see which of them are actually binding to the active site of the protein. Okay, which is, you know, for the we have to find out which is responsible for the activity, biological activity, which is somehow leading to the biological activity of the protein, that will become the leads. Out of 100, only 50 compounds are there which are actually binding to the active site of the protein and somehow triggering the activity of the protein as well. Okay. Then what will become the drug? The molecule which is binding, which is interacting, and also modulating the activity of the receptor. All these three things combinedly will become the drug. Okay. Now here comes the steps which are involved in it. We will start with the target identification and the clinical trials will be there. And preclinical and clinical are not in our hands. Okay. So target identification, then the target need to be validated. Then we will identify the lead, that lead further need to be optimized or validated. Then the docking will be there and the simulation will be there.